Okay, class, who remembers what the most important neurotransmitter is for cognitive brain functions such as learning and memorizing?
I do, Miss McNeely, Miss McNeely.
Yes, Avery?
Glutamate.
Glutamate is the most abundant and important excitatory neurotransmitter in the nervous system. Glutamate plays a major role in the cognitive functions of the brain. These functions are essential to everyday life and include learning, memorizing, evaluating, speaking, problem solving, and much more. Before stimulus occurs, the neuron is at resting potential. At resting potential, the neuron is negatively charged on the inside due to the negatively charged ions and proteins and positively charged on the outside. Because of the permeability of the membrane, more potassium ions diffuse out of the cell than sodium ions diffuse in, meaning more positive charges leave the cell than enter it. The neuron maintains the concentration gradient of sodium and potassium with the help of the sodium-potassium pump that transports 3 sodium ions out and 2 potassium ions in.
When thinking about our example earlier, the brain is stimulated by the teacher's question. The brain then sends signals and stimulates the dendrites of other neurons in order to release glutamate in order for Avery to learn and remember what the answer is. Once the stimulus reaches the dendrites, the neuron is stimulated. The threshold is met and an action potential begins at the axon hillock. As the action potential runs down the axon, sodium channels open and allow sodium to diffuse into the neuron. As sodium ions diffuse in, the membrane becomes depolarized, making the inside of the cell become positive and the outside become negative. Right behind the sodium channels, potassium channels open and allow potassium ions to exit, making the inside of the membrane negatively charged again. The action potential jumps over the myelin sheaths from one node of RON-VA to another, which increases propagation speed.
Once the action potential reaches the end bulb, the positively charged sodium ions cause calcium channels to open. Calcium ions rush into the end bulb, attach to the synaptic vesicles, and cause them to move and fuse with the membrane. The synaptic vesicles then release glutamate into the synapse. Glutamate diffuses across the synapse and then attaches to the glutamate receptor. The attachment of glutamate to the receptor stimulates the next neuron to continue the path of the action potential.
Glutamate is removed from the synaptic cleft by EAAT, excitatory amino acid transporters. The EAATs terminate glutamate after it is released into the synapse by uptake from the synaptic cleft into neuroglia and neurons. The uptake of the glutamate terminates the transmission and the neuron returns to the resting potential.